Now let's address how gas is actually transported. Gas transport's the process of carrying gases from the alveoli to the systemic tissues and vice versa. In this section, we'll explain specifically how gas loads and unloads. In oxygen transport, about 98.5% of it's actually bound to hemoglobin, and about 1.5% is dissolved in the plasma itself. Carbon dioxide has three means of transport. 70% of it goes as bicarbonate ions. 23% of it will be bound to hemoglobin, and 7% will be dissolved in the plasma itself. Let's look at oxygen first. The arterial blood carries about 20 milliliters of oxygen per deciliter, or per 10 milliliters. 95% again bound to hemoglobin. Here's hemoglobin over here on the right-hand side. And 1.5% floating in the plasma itself. Remember that hemoglobin is a molecule specialized in oxygen transport, and there are literally tens of thousands of them in each red blood cell. The hemoglobin has 4 globin portions, 2 beta subunits and 2 alpha subunits, and each of them has a heme group, this red area here, that will bind oxygen. In the center of the heme group is a ferrous ion. It's very positively charged. Oxygen has an electronegative charge, and so it binds to this ferrous ion in the middle of the heme group. When we have hemoglobin that's fully bound to oxygen, we call it oxyhemoglobin, and when the hemoglobin is not carrying any oxygen, it's called deoxyhemoglobin. When oxygen is 50% saturated, that means that the hemoglobin would be carrying 2 molecules of oxygen. At 75% saturation, 3 molecules of oxygen. 100% saturation means that all of these heme groups are saturated with oxygen molecules. Carbon monoxide poisoning results from a competition for these binding sites on the hemoglobin. Basically, carbon dioxide binds 210 times as tightly as oxygen does to the heme group in hemoglobin. Thus, it ties up hemoglobin for a long time. In non-smokers, less than 1.5% of hemoglobin is occupied by carbon monoxide. In smokers, about 10% of the hemoglobin is going to be bound by carbon monoxide. So that means even without the damage to the lungs from smoking, just the carbon monoxide in the smoke itself is decreasing a smoker's capacity to get as much oxygen as they need. If the concentrations in the atmosphere get up to about 0.2%, this can be very quickly lethal. We've seen a lot about this on the news lately. Carbon monoxide is colorless, it's odorless, it's in cigarette smoke, it's in engine exhaust, it's in fumes from furnaces and space heaters. The fact that it's in cigarette smoke is really pretty scary when you consider that people still sometimes will smoke in their cars with the windows rolled up and children in there. People will still smoke in their house with their children in there. Secondhand smoke is a big deal when you consider the carbon monoxide content and how lethal carbon monoxide is. The oxyhemoglobin dissociation curve describes the relationship between hemoglobin saturation and the partial pressure of oxygen. As hemoglobin passes through the alveolar capillaries, where the partial pressure of oxygen is high, then hemoglobin becomes saturated. As it passes through the capillaries in the tissues, where the partial pressure of oxygen is lower, oxygen will dissociate from the hemoglobin. We lose about 22% of the oxygen carried by hemoglobin at the tissues. The curve here reflects the way that hemoglobin loads oxygen. When the first oxygen moves onto hemoglobin in the alveoli, it causes a conformational change in the hemoglobin so that the hemoglobin has a higher affinity for the next oxygen that loads. So as the second one loads, it has an affinity more for the third one, and so on and so forth till we get to the fourth one. This explains the steep portion in the center of the curve here. Once we get to 100% saturation, the curve levels off because there's no more hemoglobin to carry any more oxygen. Now let's address carbon dioxide transport. Carbon dioxide can be transported in three forms: as carbonic acid, as carbamino compounds, and dissolved in the plasma. 90% of the carbon dioxide in our blood becomes hydrated. That is, it combines with water to form carbonic acid. This then dissociates into bicarbonate and hydrogen ions. 5% of the carbon dioxide will bind to plasma proteins and hemoglobin to form carbamino compounds. Chiefly, it's going to be carbaminohemoglobin. A very small portion of it will bind to the amino groups of some plasma proteins. Carbon dioxide doesn't compete with oxygen because it binds to a completely different place on the hemoglobin molecule. It binds to some amino acids on different polypeptide chains. This way, the hemoglobin can transport oxygen and carbon dioxide simultaneously. As we'll see later, each gas somewhat inhibits binding of the other gas. The remaining 5% of CO2 is carried in the blood as purely dissolved oxygen gas. That's different than becoming hydrated and forming carbonic acid and bicarbonate. The carbon dioxide is literally dissolved in the blood, as carbon dioxide would be in, say, a carbonated beverage before you open it. The relative amounts of carbon dioxide exchanged between the blood and the alveolar air differs. 70% of the exchanged CO2 comes from carbonic acid, and 23% of it comes from the carbamino compounds. 7% comes from dissolved plasma. So this is as we exchange between the blood and the alveolar air. You notice the percentages were very different for how it's carried. But what this tells us is that blood will give up dissolved CO2 gas and the CO2 that's being carried on hemoglobin much more easily than it'll be given up from this bicarbonate ion. Okay, I know it seems like it's getting a little bit equation-y, but this is really an equation that we are going to have to know over and over and over throughout the rest of the semester. Let's look at the systemic gas exchange. We have respiring tissues and capillary blood. These pictures are going to help us a lot. As a matter of fact, they'll be the basis for our diagramming in this chapter. We know that aerobic respiration produces a molecule of CO2 for each molecule of O2 that's consumed. The tissue fluid, therefore, contains a relatively high pCO2. There's a gradient from about 46 to 40 in the blood, so carbon dioxide will naturally just diffuse into the capillary blood. Here, it's going to be carried in three different forms that we already noted. Most of it will react with water to produce bicarbonate. We can see that here. This reaction occurs very slowly in the blood plasma, but in the red blood cells, it's catalyzed by an enzyme called carbonic anhydrase. We can see that here. So it happens much more rapidly. Carbonic anhydrase will convert this into carbonic acid, which will then be converted into bicarbonate and hydrogen ions. This bicarbonate will diffuse out of the cell because it's moving down its concentration gradient, less of it in the blood. But to replace the ion that moved out, we need to have another negative charge move in. This is the chloride shift. There's a transporter that antiports the chloride into the cell as the bicarbonate moves out. This allows the reaction to continue proceeding of loading carbon dioxide into the red blood cells. A smaller portion of the carbon dioxide that diffused into the capillary blood will bind with the hemoglobin. Again, not in the same region that the oxygen binds, so we create carboxyhemoglobin. Some of it will bind to amino acids on plasma proteins to form other carbamino compounds, and 7% of it will be dissolved CO2 directly into the blood. Now, at the same time as carbon dioxide is loading from the tissue fluids onto the blood, oxygen is going to be unloading. You have oxyhemoglobin showing up at the tissues. It has a lot of oxygen on it. In fact, it's 97% saturated on average. It's going to drop off most of the oxygen because there's not much oxygen in the tissues, there's a high partial pressure on the blood, and so it'll move into the blood. However, it only drops off about 22%. That leaves it 75% saturated when it leaves the tissues. The oxygen that remains in the blood after it passes through the capillary beds is called the venous reserve. The utilization coefficient is the fact that we only used 22%. This venous reserve we'll address later because different tissues will use different amounts depending on the partial pressures of oxygen in each of those tissues. So let's revisit the alveolar gas exchange now. It should be a little bit easier to understand the details now that we understand the systemic exchange. So the reactions in the lungs are the reverse of what we saw in the systemic gas exchange, where we were dropping off oxygen at the tissues and picking up carbon dioxide. Now we have blood that has a high partial pressure of carbon dioxide, and we need to unload it. So as hemoglobin picks up oxygen, which is diffusing down its gradient across the respiratory membrane, it's being picked up by hemoglobin. The affinity of hemoglobin for hydrogen ions decrease. Thus, hydrogen ions dissociate from the hemoglobin. Now they combine with bicarbonate and reform carbonic acid, which is then converted into carbon dioxide and water, and the carbon dioxide can diffuse down its gradient back into the alveolar air. As the hydrogen ions dissociate from the hemoglobin and join bicarbonate, we now have to do the reverse chloride shift to keep everything chemically and electrically in balance. So bicarbonate is moving through the transporter into the cell, and chloride is now moving out of the cell through the same transporter. It's an antiport. This whole process allows the continued freeing up of carbon dioxide so that we can have maximal diffusion from the blood into the alveolar air. So now would be a wonderful time to get out your pencils and paper and some color and diagram this process. Take the combination of both of these figures from your text in the last couple of slides, and let's go to town and see if we can understand how this process works. This equation that we're dealing with is the bicarbonate buffering system. It's going to be integral to a lot of other chapters throughout this semester, so it's important to understand this buffering system. Remember when we were talking about the baroreceptors and chemoreceptors that are in the aortic arch and the carotid arteries? And some of them were perceiving CO2 and some O2 and some hydrogen ions. This is where the hydrogen ions come from. If there's a high concentration of carbon dioxide being transported in the blood, then there'll also be a high concentration of hydrogen ions. It's these hydrogen ions that our system is most sensitive to.